This is the 7th live interactive session of Applied Environmental Microbiology, and here we will be dealing with different topics about viruses, about protozoa, about bacteria, and different questions that you might have gone through in your week 5 of lectures. So let's check the questions. The first question is, in lichens, moisture is trapped by, and the options are fungal hyphae, growing media, algal cells, and humans. You have to let me know what is the correct answer. Option 1 is the correct answer. Let's see. Yes, option 1 is the correct answer. Because, what do you think option 1 is the correct answer? Sorry. Yes, can you please repeat? Actually, there is a lot of noise in your background, but I think what you wanted to say is that fungus is attached with lichens. That's what you said, right? What do you think is a symbiotic relationship happening between which two organisms in lichen? Does anybody know that? It's fungi, and what is the other symbiotic organism? Neha, are you aware of that? Yes, sorry, can you please repeat? No, sorry, you are wrong. Anybody else? Let's discuss. So, what is a lichen? A lichen is basically not a single organism. It is a symbiosis between different organisms, and mainly it is symbiosis between one fungus and another one can be an algae or a cyanobacteria. Now, this non-fungal partner, that is the algae or the cyanobacteria, should contain chlorophyll, and it is called photobiont. Whereas the fungal partner is referred to as mycobiont. Now both this partner or the lichen consists of, as I said, one mycobiont and one photobiont. And however, that is not universal. Sometimes the lichen can have more than one photobiont, that is more than one algae or cyanobacterium as a partner. Now this algae photobionts are called phycobionts, whereas cyanobacterial photobionts are called cyanobionts. So, if we look microscopically inside this lichen, we will see that this fungal partner is composed of filamental cells, and each of this filament is known as hyphae. Now, this hyphae grows and they extend their branches and they form a structure which is composed of clusters, and they look more or less like globose cells. On the other part, there is a part of this lichen which is not involved in reproduction, and it is known as the body or the vegetative tissue of the lichen, and it is called thallus. So this thallus form is different from the fungus or algae that is growing separately. So it is made up, this thallus is made up of the hyphae of the fungus. So this is the picture of the lichen that you can see, different types of lichens. So these lichens are generally found around moist areas. So let's see how this symbiotic relationship exists in the lichen. Now if you look at this diagram, this shows a cross section of the lichen. So this portion is the cortex and the outer layer of tightly wound fungus filaments, that is the hyphae. Okay, this portion. Now this is followed by the photobiont layer, which can have the cyanobacteria or the algae. Next is the loosely packed hyphae, and it is known as medulla. And then comes the tightly woven lower cortex, and finally there is anchoring hyphae, which is called rhizines, which attaches this whole thing to the substrate. Okay, so now the question was, which part of the lichen actually is responsible for absorption of moisture? Now let's find out which part is responsible. So for that we have to study the symbiotic relationship of that goes on in the lichen, that is what functions are performed by the fungus and what functions are performed by the cyanobacteria. So let's check out that. So a lichen is, as I said, is a composite organism, and it emerges from algae or cyanobacteria living among the filamentous hyphae, and then they are mutually always in a symbiotic relationship. Now what scientists observe is that the fungi benefit from the carbohydrates that is produced by the algae or the cyanobacteria during the photosynthesis, whereas the algae and the cyanobacteria, they benefit by being protected from the environment by the filaments of the fungi. And at the same time, these fungal filaments also provide moisture and nutrients from the environment, and they also provide anchor to the environment. Okay, so the major function of the fungi is to provide moisture, nutrients, and also provide an anchor, whereas the algae and cyanobacteria, the main function is to go on with photosynthesis, which provides carbohydrates to the fungus. Now sometimes these photosynthetic partners, that is the cyanobacteria or the algae, they can survive outside of the lichen, but what is observed is that the lichen can then extend the symbiotic association, and it can go on to the ecological range of both the partners. Okay, and now therefore sometimes scientists also saw that both the partners sometimes gain water and mineral nutrients from the atmosphere, that is from rain, dust, etc. And the fungal partner protects the algae by retaining water and serving as a larger capture area for the mineral nutrients, because it is spread around because of its hyphae. So it has a larger area to capture the mineral nutrients. Also it provides the minerals that are available in the environment. Now if a cyanobacteria is present as a primary partner or another symbiont in addition to a green algae, as in certain tripartite lichens, then they can fix nitrogen. So cyanobacteria, if it is present as the photobiont, then it can fix atmospheric nitrogen and it can complement the activities of green algae. Now in these three different lineages, the fungal partner has lost the mitochondrial gene, that is called ATP9, and its key function is mitochondrial energy production, and therefore this fungi is completely dependent on the symbionts or the phycobionts. Now both this algal and the cyanobacterial cells, being photosynthetic, they reduce atmospheric carbon dioxide into organic carbon sugars to feed both the partners. And the sugars can be used to produce alcohol like ribitol, sorbitol and erythritol, which is absorbed by the microbiome, that is the fungus. Cyanobacteria also are known to produce glucose, which can be used as a food source, whereas the fungal cells make the photobiont leak out the products of photosynthesis, so they can be absorbed by the fungus. So by now, if you have to tell in nutshell, we learned that lichen is not a single organism. It is an organism which has two parts. One can be a fungus, which is called microbiont, another can be a cyanobacteria or a algae, and that is called photobiont or phycobiont. Now, what does the fungus do? The fungus provides anchor, it provides nutrients by absorbing it from the surrounding because of its long hyphae. Also, it absorbs moisture. And what does the algae and the cyanobionts do? They produce food by the means of photosynthesis, which is absorbed by the fungi. And that is much more required because the fungi lost the mitochondrial gene, which enables it to, or which does not help the mitochondria or does not help the fungi to produce energy with the help of mitochondria. So therefore, as I said, the correct option is lichen's moisture is trapped by fungal hyphae, okay, whereas algal cells performs photosynthesis. Now coming to the next question. Several environmental species of hydrocarbon-eating bacteria like Alcanovorax borcumensis feast on spilled oil, assisting with disaster cleanup efforts. This is an example of, your options are bioterrorism, biomedication, bioremediation, biohazard. Tell me, what do you mean by bioterrorism? Okay, so bioterrorism is the deliberate release of viruses, bacteria, toxins or any other harmful agents which can cause illness or death to the people, and it can be people, animals or plants. And this type of terrorism generally happens during war, and the biological agents that are spread, they can be through air, water or the food. So who does this kind of terrorism? So basically these are done by terrorists. They choose a particular kind of organism which can be a smallpox virus and or an anthrax, and they spread it from person to person. So this has happened previously in World War I, and it is very detrimental for the human health of the countries in war, and therefore WHO had intervened and they had ordered that this should not be the case, and bioterrorism was banned. But still it is a threat to the environment, and still it is a ongoing threat. So clearly bioterrorism is not the answer to this, because this example is basically telling us that something good is happening because of the bacteria eating up or feasting up on the oil. So can we check on what biomedication is? Can anybody tell me what they know about biomedication? Sorry, can you please repeat? Yes, the correct answer is bioremediation. Can you tell me a bit about bioremediation? Yeah, it can be not just water but also environment. Okay, let's check the definition. So bioremediation, it is a biotechnical process by which the microbes are used to clean up contamination in the environment, and therefore it can also be said to be a type of waste management technique. And the organisms that are involved in this technique, they remove or utilize the pollutant that is present in a pollutant area. Now if you are to see that there are several remedies when we look at contaminated water or solid or tip treatment. Like if we see that generally there are different kind of contaminants present in land, in water, and that is released from the industries. There are in water we see oil spillage happening. Sometimes the industries release toxic water in the and which has got presence of high concentration of ions in it, or heavy metal ions in it, and this is where bioremediations come into play. And bioremediation is basically using microorganisms to basically metabolize all these toxic chemicals in order to produce a non-toxic product which is not detrimental to the environment. Okay, so what kind of microorganisms are used? So microorganisms like bacteria and fungi, they play a major role in this kind of bioprocess, and they in this process what they do, as I said, they break down the waste into nutrients and organic matter which are no longer toxic to the environment. And this process is a very efficient process of waste management, but the only problem is that it cannot destroy 100% contaminants, and it is also a very slow process. This is because the bacteria cannot they cannot easily digest contaminants like chlorinated pesticides, or they take some time to clean up oil spills. But at times it is seen that when the same microorganisms are used to destroy heavy metals like lead and cadmium, they take a lot more time and not 100% is removed. So there are different kinds of bioremediation. So the first type is biostimulation. So what happens in this case? A bacteria stimulates, or to say it initiates the process wherein a contaminated soil is mixed first with the special nutrient or substances which has the vital components which can be either in liquid or gas, and then that stimulates the growth of the microbes, which results in an efficient and a quick removal of contaminants by the bacteria or by a group of bacteria. Next type is bioaugmentation, wherein there are certain sites where the microorganisms are required to extract the contaminants. For example, what this kind of treatment happens in municipal water treatment plant. In these cases, sometimes bacteria is used to clean a particular part of the or particular waste present in the water. But the only problem here is that the growth of the microorganisms it's not possible to control the growth of the microorganisms. Okay, now the third type of bioremediation is intrinsic bioremediation. Now in this process of bioremediation, it is most effective in the case of soil and water, because these two biomes have high probability of being full of contaminants and toxins, and they produce and in this kind of place it is difficult to detect kind of leakage and contaminants and toxin, and they can find their way to the lakes and they can contaminate petrol. So this microorganisms can remove all this kind of toxins and they can clean the petrol tanks. Now there are certain limitations of bioremediation. The first limitation is that not everything can be biodegraded, like heavy metals, radionuclides. These are elements that cannot be biodegraded, or they cannot be like, if needed, they can be biotransferred into less mobile forms or less toxic forms. Okay, so and the biodegradation requires microbial population with certain metabolic capacity to degrade the pollutant, and therefore this process becomes highly specific to a particular microbe. And also we have to take care of the environmental factors that is required for that particular microbe to thrive, and we have to regulate the environmental conditions in that way. And sometimes small scale laboratory results that scientists work, they cannot be manifested on large scale results, because in laboratory there is control of environment, whereas in the environment, when in the practical environment or practical scenario, it is very difficult to hold control over all the other environmental factors. Okay, so therefore these are some limitations of bioremediation. Even though it is the most inexpensive and the most best used technique, but still it comes with certain limitations. Now coming to the next question. Plants get infected by viroid by both acquiring the infection from infected neighboring plants by air and by coming in contact with the infected cells. Now you have to tell me if this statement is true or false. Yes, the correct option is true. So any of you know what is viroid or viroid? Anybody know what is viroids? Yes. So as Neha clearly mentioned, that viroids are small single-stranded circular RNA which are infectious pathogen. They are naked RNA. And why are they called naked RNA? Because unlike viruses, they do not have a protective coating around them. So where do they stay? They inhabit angiosperm, that are flowering plants, and they cause diseases which actually cause a lot of economic problems to the humans because they kill the plants. And this nucleus of the virus or the RNA of the viroids is single stranded circular RNA, and the length can vary from 246 to 463 nucleotides, and they are strictly found to infect only plants. So about now 30 viroids have been characterized, and 28 of them are known to infect dicotyledonous, that is double seeded plant, whereas other two infect monocotyledonous, that is single seeded plant. Now let us see what are the two main families of viroids. One is Pospiviridae. So members of this family are named after the potato spindle tuber viroid. So this viroid has got, or viroid has got, a rod-like structure with a small single stranded region, and they have got a conserved region which is centrally located, and they replicate in the nucleus of the host. Now the plants that are infected with this family members, the RNA of the viroid, they get imported into the nucleus and they are copied by the plant DNA dependent RNA polymerase, and then this viroid is copied by a rolling circle mechanism which produces complementary linear and concatenameric RNAs. So what happens is that inside the host, that is inside the plant, they actually replicate their genetic material to form concatemers, that are long chains of RNA. Now these are copied again and again to produce such concatenameric linear molecules, and then they are cut by the host enzyme RNA polymerase 3, and they are joined by the host enzyme to form circles, which again becomes a new viroid particle. So this is how viroid replication happens for Pospiviridae family. Now the next family is Upsonviridae. So this was named after avocado sunblotch viroid. So this viroid also has a rod-like branched region. However, they lack a central conserved region which was present in the case of Pospiviridae, and they replicates in the chloroplast of the host. Now these RNA molecules are they are ribozymes, and they have got enzymatic activity which is crucially required for RNA replication. Now in the plants, the members of this kind of family, this viroid RNA is imported into the chloroplast, wherein the complementary concatenameric RNAs are produced, and then they are cleaved, cut, and then they are ligated by the ribozymes, which are encoded inside the viroid RNA. So in this case the host machinery is completely used, but in this case a host machinery is partially used, and after cutting the cutting is done by a ribozyme. Here in Pospiviridae the cutting and the joining is done by RNA polymerase 3. Okay, so there is a major difference. Now the question comes that how does this viroid spread? So viroids, like viruses, are contagious pathogens, and they are easily transmitted from one crop to another crop. And the most common reason is that one plant can get in contact with another infected plant, and then they can acquire these viroids. Okay, so therefore the option that was there, that plants get infected by viroid by acquiring the infection from infected neighboring plants via air, yes, it is true. Or the plants can get infected by coming in contact with the infected cell. So yes, that's a true statement. Now coming to the next question, match the following. So your options are archaea, bacteria, protozoa, and the second column has eukaryotic, ubiquitous, or present almost everywhere. Next option is associated with extreme conditions. You have to let me know what is the correct option. Yes, option 3 is the correct answer. So, can anyone tell me what is a protozoa? Yes. Can you give an example? Yes. So, protozoa is unicellular eukaryotic heterotrophic organism, and they are free living or they can be parasitic. There are many species of protozoa. The main characteristics of protozoa is that they lack a cell wall, and they have different types of cell organelles that performs various tasks which is important for the or tasks that are performed by higher order animals. For example, it has got a mouth, it has got a anus, it has got a mouth over here, as you can see. It has got food vacuoles over here. It also has got a cilia with which it beats to move. Also, it has got anal pore, and all this shows that protozoa has cell organelles that can perform the tasks that are performed by different organs in higher animals. The examples of protozoa can be Plasmodium, that is a malarial parasite, Trypanosoma, which causes sleeping sickness, and Trichomonas, that causes trichomoniasis. Okay? Therefore, matching protozoa with eukaryotic is the right option, and matching bacteria with ubiquitous, that is present almost everywhere, is also the right option, because we have studied that there are certain bacteria which can survive even in high salt condition, even in high pressure condition, even in normal conditions like soil, water, etc. And therefore we can say that the bacteria are present ubiquitously, that is present almost everywhere. Now, who else remembers about archaea? We had discussed in one of our previous classes in details. So, can anyone tell me what is one of the special features of archaea? Yes. But the main characteristic is that they can survive in extremophilic conditions, right? And they have features that are similar to both eukaryotes and prokaryotes. So again I will revise it. So archaea can be obligate or facultative anaerobes, that is they can flourish in the absence of oxygen, and they can also undergo methanogenesis, which is a way in which they can metabolize methane. Cell membranes of archaeobacteria are mainly composed of lipids which help them to thrive in adverse conditions. The rigid cell wall provides shape and support to archaeobacteria, and it protects it from the severe conditions that can be hypotonic conditions. Cell wall of archaea is also composed of pseudomurine, which prevents the archaea from the effects of lysozyme. That is an enzyme that is present by the immune system of the host. It also dissolves the cell wall of the pathogenic bacteria and dissolves the cell wall of the pathogenic bacteria. Also, the archaea has this membrane-bound organelles, like it does not possess membrane-bound organelles like nuclei, endoplasmic reticulum, which is also the same case for bacteria. So any double membrane bound organelles, that is nuclei, endoplasmic reticulum, mitochondria, lysosomes or chloroplasts, they are not found. They can live in a variety of environment, and therefore they are called extremophiles. That is, they can survive in acidic and alkaline and aquatic conditions, and also sometimes in temperatures that are above the boiling point. Also, they are seen to withstand high pressure of about 200 atmospheric pressure, and therefore archaea are believed to have been in the world when the world was in its premature stage. That is, when the water or the current environmental conditions was not predominant, when the earth was in its formative period, then archaea is believed to have developed. Therefore it can still withstand such harsh conditions, and due to its withstanding such harsh conditions, it has developed certain features which help them to withstand these conditions. Now coming to the next question, algae are photosynthetic eukaryotes. You have to tell me if it is true or false. It is very easy. Yes, it's a true statement. So over here you can see a picture of algae. Most of you can see this kind of organisms growing because of the presence of moisture in it, in the environment. Just hold on a second, I'm getting a call. So basically an algae is defined by a large group of eukaryotic photosynthetic organisms, and they do not share a common ancestor, and therefore we cannot say that they are related to each other. So they are polyphyletic. Now algae can be multicellular, for example giant kelp and bound algae, and sometimes it can be unicellular, that includes diatoms, euglenophyta, dinoflagellates, etc. As I said, you can find this organism in a moisture rich environment. So it requires moist and watery environment, and therefore they are generally present near or inside water bodies. So they are similar to like land plants, they grow, they also are responsible for producing the maximum amount of oxygen that is released into the world's atmosphere with the land plants. And the difference with the land plants is that they lack any structural component which are generally present in the plants. For example, stems, shoots and leaves. And also they do not possess vascular tissues which help them circulate essential nutrients throughout the body. Now coming to the main characteristics of algae. So the main characteristic is that it is photosynthetic and it can exist as unicellular or multicellular organism. Algae has a it lacks a well-defined body and the structures like roots, stems, shoot, etc., which are present in higher order plants. They are found where there is adequate or excess amount of moisture, and the reproduction in algae can happen in both asexual and sexual forms, and asexual reproduction occurs by spore formation. Algae can also be free living, but sometimes they can be associated with a symbiotic relationship with other organisms, and the example is, can somebody give me an example? We just read about it in the first slide. What is the symbiotic relationship shared by the algae with other organisms? Can anybody tell me that? Lichen is an example. So algae thrives in a symbiotic relationship with fungus in lichen. Otherwise algae can also be free living. So there are different types of algae. One can be red algae, and it is also known as rhodophyta, which is a distinctive species that are generally found in marine as well as freshwater ecosystem. So they appear red because they have got pigments, that is phycocyanin and phycoerythrin, which gives them this color. Sometimes green coloration is observed if chlorophyll is present, but this kind of algae, they lack chlorophyll B or beta carotene. The next type is called green algae. They are also large group of algae having primarily photosynthetic pigment, that is chlorophyll A and chlorophyll B, along with other auxiliary pigments like xanthophylls and beta carotene. So certain higher organisms use this kind of green algae to conduct photosynthesis for them. The members of this green algae can be unicellular, multicellular, colonial, and some can be flagellates, and the example involves Spirogyra, Ulothrix, Volvox, etc. The next category is blue-green algae. So blue-green algae was one of the most well-known type of algae, but since they are prokaryotes, they are not currently included under the category of algae, because algae is a eukaryotic organism. So blue-green algae is also known as cyanobacteria, and these organisms, as any other algae, will survive in aquatic environment, and this can include river reservoirs, creek, lakes, oceans, etc. They obtain energy through the process of photosynthesis, and they also are able to do nitrogen fixation. For example, nostoc, anabina, etc. So we got a brief idea of what algae is. Now coming to the next question. With regard to PRNP gene discussed in the lecture, PRPSC misfolded protein are regarded as pathogens because This mutates the genetic coding of the cell. You have to tell me if this statement is true or false. What is PRNP stands for? Or what do you know about PRNP? Yes, PRNP means a prion protein. So PRNP gene, actually I will go to the answer. The answer is false. It is not due to a mutation in the gene encoding the cell. I'll tell you why. I'll gradually proceed. So let's discuss about what is PRP or what is known about it. So basically PRP is prion protein and it is encoded in the human by the PRNP gene. So PRNP is the name of the gene and the protein is called PRP, that is prion protein. And this gene is also known as CD230 cluster. So this protein is predominant, that is a prion protein is predominant in the nervous system, but it can also occur in tissues throughout the body. Now this human prion protein producing gene is located in the short T arm of chromosome 20 between the end terminus of the arm and position 13 from the base pair of 4615068 to base pair 4630233. Now this gene provides instruction for making a protein called prion protein. This is what we have discussed about. Now when this protein is active in the brain, they are proposed to do several kind of important processes. They are known to transport copper into the cells and protect brain cells or neurons from injury. It has also been shown by studies that PRP helps in the formation of synapses, that is a junction between the two nerve cells through which a signal passes, and therefore it helps in cell-to-cell communication. So this prion protein that is present in human, when it is in its normal form, then it can do the following functions as I mentioned. But scientists observed that this protein can exist in multiple isoforms. One can be the normal form, which can be coded as PRPC form, and another can be a protease resistant, or that is PRP resistant disease causing protein, that is PRPSC, where SC stands for scrappy, and another is an isoform that is located in the mitochondria. Now let us learn what is the difference between PRPC and PRPSC, that is a diseased form. So let's first check the properties. So the properties is that for PRPC, it is the normal category of the protein that is produced by the normal PRNP gene, but PRPSC is pathogenic. Now PRPC is not protease resistant, but PRPSC is protease resistant, and they have a stable core containing a residue of 90 to 231. PRPC is generally located in the plasma membrane, whereas PRPSC is located in the cytoplasmic vesicles. PRPC is known to be soluble, whereas PRPSC is insoluble, and the PK sensitivity of PRPC is highly sensitive, whereas PRPSC is partially sensitive. Now this PRPSC is detrimental if produced in the humans because it can be responsible for several neurodegenerative disorders. Okay. And why so? Because both of them have different structures. As I said, PRPSC is a isoform. That is, it is a conformational isoform of PRPC. But the orientation is different. So what is the abnormality in PRPSC? It is seen that it has different secondary and tertiary structure as compared to PRPC, even though it has got identical primary sequence. So if it has identical primary sequence, then obviously there is no mutation in the gene. Everything is caused in the arrangement of the protein or arrangement of the amino acid or its hierarchy. The protein folding is where this particular pathogenic protein is developed. So if you look at PRPC, you will see that it has got 45% of alpha helices, whereas 33% of beta sheet, that is more of alpha helices, are present in PRPC. But if you see PRPSC, it has got 30% of alpha helices, that is alpha helices substantially decreases, but beta sheet percentage increases in PRPSC, as you can see in the figure. Therefore, this folding of PRPSC is responsible for making it resistant to proteolysis, and the accumulation of PRPSC will cause more PRPC to transform into PRPSC. Therefore, it leads to more accumulation of this protein, which actually is responsible for the neurodegeneration. Okay, so therefore, as I said, this statement is false because there is no mutation happening in the gene itself, but the folding of the protein is wrong, and therefore that leads to a formation of a pathogenic protein. Let's go to the next question. Which factor allows rhizobial cells to invade the root cells in legumes? The options are rho factor, alpha factor, NOT factor, beta factor. Can anybody answer? Yes. The correct answer is NOT factor. So we had discussed in the previous class that NOT factors are special molecules that are excreted by the rhizobia, and they majorly determine the range of hosts, the infection and the nodulation they will do in the presence of the leguminous plants. And as I said, how does this node develop? We have discussed in the previous classes. That is, initially there will be one or two bacteria going in through the root hair, and then they will form a inoculation loop, and this is where the bacteria will be internalized, and this will be then at this position there will be several division of the cells which will form a nodule like structure. And this is where, since the bacteria is accumulated here, this is where the bacterial nitrogen fixation will take place. Okay. Now coming to the next question. When a bacteriophage infects the host cell, which of the following will enter the host? Your options are outer part of the virion, protein coat, capsid made of capsomers, host genetic material, all of the above, none of the above. You have to tell me the correct answer. Neha, did you answer? Sorry, I could not hear. Please repeat the answer. All of the above you are saying? Which one are you saying? Fifth or sixth one? Is option 5 correct? or option 6 correct? 5? No, you are wrong. So the correct option is none of the part enters into the bacteria. I'll show you why. So first, for that, let us understand what a bacteriophage is. Okay, before I explain, can anybody tell me what a bacteriophage is? Okay, I will explain. So bacteriophage is a virus that infects bacteria. So as if we go by structure, then that you can see that it has got it can be of different types of shapes. So generally this phage contains DNA or RNA as its genetic material, and this DNA or RNA or genetic material has few genes, and it can range from few genes to few hundred depending on if it will exploit the host machinery for its replication. So basically this bacteriophage has got a capsid which has got an icosahedral symmetry, or it can be filamentous in shape, or it can have head or tail shape. So over here you can see the three different types of, or the three different physical characteristics of the bacteriophage. Okay. Now, how does this bacteriophage cause infection and who do they invade? As the name suggests, bacteriophage infects bacteria, and they reproduce by going through two kind of cycles. One can be a lytic cycle, or by lytic I mean lysis of the bacteria, and another can be lysogenic cycle. Okay, so what happens in a lytic cycle? So in a lytic cycle, if you see, the first step is attachment of this bacteriophage. Okay, so what you can see over here, that a live bacterium is here which is having a bacterial chromosome. The bacteriophage attaches, and you can see here is the head, this is the tail. So the proteins in the tail of the bacteriophage, they bind to the specific receptor which can be a sugar transporter on the surface of the bacterial cell. This leads to the attachment of the phage. Next step will be the entry of the phage. But what does that entry mean? Will the entire part of the phage go in? No. As you can see over here, only the genetic portion of the phage will go in. But over here, nowhere in the option genetic material was written. Outer part of virion is drawn. Correct answer. Protein coat is not correct answer. Capsid is also not correct, because all these are outer coat of the virus which does not goes in. Host genetic material is again wrong, because that is already present in the host. So therefore, if this was viral genetic material, then this option would have been correct. So the next step is entry, wherein the virus as a whole does not enter, only the genetic material of the host, of the virus enters into the host. Okay, so basically the bacteriophage or the virus, they inject the double-stranded DNA genome into the cytoplasm of the bacteria. Now what does the viral DNA do? So inside the host, the viral DNA will be copied, and using the host machinery, because this replication of the viral DNA will produce different kinds of proteins which are responsible for the assembly of these phages again. That is, it will produce more and more copies of the viral DNA, and it will produce more and more copies of the capsid proteins. Now once it has done that, it will go on to assemble this DNA inside the capsid proteins. That is, the capsid assembled from the capsid proteins, and they are stuffed with DNA to make a lot of new phage particles. This will be followed by lysis of the phage from the bacteria. That is, in this step, the phage will make small holes on the host cell, which will cause lysis of the bacterial cells, and the new phages will just come out. So this lysis releases 100 of new phages from one host bacteria, and few cycles of this infection can let the phage spread like wildfire among the bacterial population. Now, as I referred, that there are two processes by which the phage can reproduce inside the bacteria. One is this lytic cycle, which I discussed here. The next step is the lysogenic cycle. Now what happens in this cycle? In this cycle also, the first step and the second step is same. That is, there will be attachment of the phage, which will be followed by entry of the viral DNA into the host. But the next step is slightly different. That is, there will be integration of the viral DNA into the chromosomal DNA of the host. And now this viral DNA will stay inside the bacteria, and when the bacteria goes into division, it will replicate the viral DNA along with its own DNA. So when there is a need, or when the bacteria is immunocompromised, then this viral DNA can produce bacteriophages, which can be released more in numbers because the bacteria is also dividing. Okay, so these are the two ways in which the the virus can replicate in its host bacteria. So I hope this answer is clear, why it is not all of the above, but it is none of the above. Okay, with this we end our live session. If there is any question, you can ask. Is there any question? Okay, then we can leave the live session.